All right, I think I'm gonna get started. So first I'd like to introduce my committee members. My primary advisor, Eric Selker, who has guided me throughout this entire process and been a mentor and advising the direction. David Garcia, my second reader, who works with Eric frequently and has a lab right next door to ours, so I see him often. And Barbara Mossberg, my Clark Honors College advisor, who's just been lovely throughout this entire process. And I'm going to be presenting my thesis, the induction of histone H3K27 methylation by non-native telomere repeats in the organism Neurospora crassa to you all today. I've been working on this project for quite a long time, and I'm excited to present it in partial fulfillment of my biochem major and the Clark Honors College. All right, so let's just jump right into it then. So human development is amazing. The fact that a single cell can grow into a being as complex and adaptable as a human, from an embryo to finally a fully formed human being, is just incredible. Like, the fact that we're all alive is amazing with how much has to go right. How the heck do we do that? To accomplish this and to keep on living, our bodies need to constantly orchestrate a symphony of regulatory processes to have the right materials and molecular machinery at any given moment. And all of that is coded in our genetic instructions, our DNA. Most of you will be familiar with a typical chromosome.
Can I stop you for 1 second? Would
Yes.
you record this? Because
I am currently recording
we're
this.
going to really want this. Okay, thank you. Thank you.
There on the left, you see a chromosome, as you would if you looked into a microscope. And if you zoomed in a little bit further, you'd see the genes. With a magical microscope, you'd see all the genes that are there a little bit further to the right. And you can see that relatively short region of just one chromosome has an absolutely massive amount of different genes that code for many different functions. And a problem emerges, like, this obviously has to fit somewhere, right? And the average human cell is only about 100 micrometers across, which is, can't see that with the naked eye. And our DNA, if you stretched it all out, would be almost 2 meters long. So that's a bit of a problem when you want to have a copy of this DNA in every single one of your genes, or every single one of your cells. So how do we fit it all in there? The answer that our bodies have come up with, and many organisms is the formation of chromatin. Broadly, chromatin is a way of packing and condensing DNA into manageable sizes. Similar to how when you sew, you don't have your string cascading about your body, you wind it on a spool. And in fact, your body has protein spools that it uses for DNA, which are a complex of 8 proteins called histones, that the DNA is wrapped around, forming something called a nucleosome. And when wrapped around the nucleosome, we call it a beads on a string formation, as you see there near the top. It looks like, just little beads cascading on a string. And if it's wound a little bit tighter, you can get further structures and further packing into a theoretical structure called the 30 nanometer fiber, or in general, heterochromatin, which, as you can see, it's a lot more densely packed. There are a lot more proteins per length of DNA, and you can imagine it would be harder to access any given piece of the DNA. And indeed, heterochromatin is usually found near silent genes and genes that are minimally transcribed. So how does the body decide which bits of the genome need to be tightly packed and which needs to be freer to be transcribed and make the machinery that we need at a given moment? Well, one of the ways we regulate chromatin is through histone modifications facilitated by many different complexes of proteins. Of particular interest to me is the polycomb repressive complex 2, as you can see in this image. It's associated with the deposition of methylation on lysine 27 of histone H3, which is one of those 8 histone subunits that make up the nucleosome. The catalytically active subunit is SET7 in Neurospora, or EZH1 as it is named in humans. As you can see in the image, it is putting methyl groups onto a histone tail. H3K27 methylation is typically associated with gene silencing and general like transcriptionally silent chromatin. Many other proteins have been found to associate with this core in a variety of different circumstances and organisms, but what you see up there is the core unit. Misregulation of this complex, like if you just methylate random things, can cause cancer, as you might imagine, as you wouldn't have the right genes at the right time, or just transcription goes wrong. And that, that can lead to cancers like leukemia and things like that. So like horrible illnesses. You can kind of, methylation deposited by PRC2 is a type of epigenetic marker. And epigenetics is the study of mitotically and or meiotically heritable changes in an organism brought about by the modification of gene expression without altering the genetic code itself. So you can also look at the roots of the word epi, meaning on top, and genetics, meaning genetics. So these are modifications that are placed on top of your genes. You can think of it like a complex series of switches for the genome that can be turned up and down in response to a wide variety of stimuli. How complex is this, you might ask? Well, if we look at only the lysine methylations on histone H3, of which there are 19 potential sites, there are over 250 trillion permutations for just one of these histones. And given that there are possibly 30 million of these nucleosomes, let's see, where am I going? 30 million of these nucleosomes in any given place, you can see how it's just mind-bogglingly complex. Like there's no way, like it's incredibly intricate, and it's amazing that our body can regulate this at all. And so aberrant modification of these epigenetic markers can lead to, again, aberrant gene expression and cancer. So therefore, it becomes important to know how the body regulates this complex genetic switchboard so we can understand what can go wrong and eventually how we can fix it. The current body of literature on epigenetics is growing every day. However, there are still vast gaps in our knowledge. Particularly, I seek to help characterize how PRC2, how and subsequently H3K27 methylation is regulated. A good first question would be, where exactly is this H3K27 methylation found? One place this mark is commonly found to be enriched, at least in Neurospora, is near the DNA telomeres. Now, telomeres are found at both ends of chromosomal DNA and perform a function, as I have up on the screen, similar to the plastic caps or aglets of your shoelaces. They keep your DNA from fraying as it is used, during DNA replication in particular. Fraying in this case wouldn't be as simple as going on to Target and buying a new pair of Converse. It would lead to DNA damage, chromosome catastrophe and death, and cancer. But like, it's not good. Telomeres are typically many repeats of a short sequence of DNA in vertebrates and a few other organisms, including us. This is TTA-GGG, and that's just repeated in humans thousands of times, and in other organisms more or less. But all organisms that have linear chromosomes need some form of protection near the ends, or their DNA gets damaged. So this is just what we chose to use, or rather most eukaryotes do. And let me bring up the methylation patterns here. So that's a chip sequencing assay, which essentially is where you count the amount of times a mark is on a certain region of DNA. I'll talk more about it later, but take my word for it for now. But you can see there are peaks of this methylation where it is more frequently methylated near either end, right near the telomeres. And this region we call the subtelomere region, which will be relevant later. Subtelomere just meaning it's right next to the telomeres, but not on the telomeres. Let's see. So why does that happen? Why there? Why near the telomeres? The chip data was, right here, is collected in my model organism, Neurospora crassa, which I use to help figure out why this happened. Neurospora is a relatively simple eukaryotic organism, a filamentous fungus that somewhat resembles the popular drink from the 90s, Tang. It has chromosomes flanked by telomeres and exists in a haploid state. It's a great model for studying protein methylations in particular, or histone methylations rather, because it's very easy to propagate in the lab. You don't need to have rat cages or ethics boards about it. They provide lots of options for manipulating the genome due to its high genetic and biochemical tractability, so you can cut and paste genes all over the place. Its genome, by the way, is small and fully sequenced, which allows us to do some fancy stuff. It has a copy of PRC2, as you saw on the previous slide, and H3K27 methylation is still a marker for silent genes, unlike some other simple model organisms such as budding yeast. And most importantly, perhaps, hyper and hypomethylation of the genome does not result in gross developmental errors. So there may be some slight defects, maybe errors in sexual performance or things like that. But in general, the organism doesn't die, which means I can study methylation patterns that in other organisms would just kill the animal and you wouldn't get to observe it. So this research was inspired by a paper out of the Sulker lab from 2018, where DNA telomeres were found to be sufficient to induce and necessary for the maintenance of subtelomeric H3K27 methylation. Furthermore, you can see that internal telomere repeats were sufficient to induce ectopic H3K27 methylation. If you look at the chip tracks on the bottom there, you can see the wild type strain without the telomere repeats inserted into the internal region of the genome. There's absolutely no methylation or just background around this region. But right when we put it in green there, you see robust methylation for hundreds of thousands of base pairs around the insert site. And just to give you context on what ChIP-seq actually is, suppose the genome, imagine the genome is like a highway, right? And you want to know the location of every car on the highway, or every yellow car in particular. ChIP-seq takes a global snapshot of that whole highway, so you can tally up the locations of all the yellow cars and plot where exactly they are in like a little histogram type thing. In the real assay, you would use an antibody to determine the type of car or protein you're looking for in this case. And in this case, we use an antibody against H3K27 di and trimethylation, which gives us the transcriptionally repressive markers. So what about this sequence? The TTA GGG is able to induce H3K27 methylation. Like, it's weird. Like, there are plenty of sequences in the genome that somewhat resemble this. Granted, no repeats very often, but they don't seem to be important for the induction. So why does this repeated sequence of telomere repeats tend to induce methylation spontaneously? Well, to that end, I suspected that the ability for it to form a structure called G quadruplex was our culprit. Guanine quadruplex, or G quadruplex, is a DNA and RNA secondary structure formed by G-rich strands that have 4 or more repeated sequences of at least 2 guanine bases, or from up to 4 different sections of DNA, but in the case of telomeres, it would just be the same strand of DNA. 4 Gs form into something called a D tetrad, as you can see in a figure A up there. They pair a little weirdly, like weirder than normal, around a cation. But it's when you have consecutive Gs in repeated sequences that those tetrads are able to stack and form like a super stable structure that forms quite predictably, and you can calculate what sequence of DNA are likely to form this. In principle, G quadruplex can form in many ways from a variety of organisms, which is what you see in B. These are just a few ways that from a single strand G quadruplex could form. And let me pop the telomere sequence up there. So that's the telomere sequence. You can see that those Gs are participating and would participate in a G quadruplex, sort of like that. And the location of G4 forming regions are often non-random and correlate with functionally important regions of the genome involved with replication, gene regulation, and telomere maintenance. The human and Neurospora telomere sequence is known to form G quadruplex in vivo. That means inside an organism. And it was found that PRC2 has a higher affinity for the RNA sequences that can form G quadruplexes over those that do not. Therefore, it becomes likely that G quadruplex may form some kind of role, if only just to recruit the complex to perform its catalytic activity. Other organisms also, even ones with different sequences of telomeres, also can form G quadruplex, such as Arabidopsis and Tetrahymena. So to determine whether non-native telomere repeats could also induce ectopic H3K27 methylation, I designed a series of DNA constructs to insert into Neurospora at the CSR1 locus, which I'm going to pop up right here. So you can see the wild type sequence up at the top there, GTA, GGG, and I have the differences in red. First, I made a negative control, which just has a C in the middle, which interrupts the ability to form G quadruplex, because again, you need consecutive Gs for that stacking to happen. And the C was chosen to maintain the GC percentage, which has been known to influence, or generally there's a trend that heterochromatin regions are more AT rich than others, so I didn't want to bias that if I could avoid it. Arabidopsis, which has an extra T in the base, but for the most part is the same, so it's a 7 repeat motif. And the Tetrahymena telomere, which is TTGGGG instead of, so just an A swap to a G, which theoretically should be able to form a stronger G quadruplex, but you know, who knows? So my experimental design generally went making these DNA constructs and annealing them together, as you can see on top. And since I was targeting the CSR1 locus, I needed to insert the DNA into a targeting plasmid. So what you do is I designed these constructs with restriction enzyme sites, which are basically little molecular cutters for DNA that make ends that are unique to that cut site. So mismatched ends won't pair back together. So I cut both the plasmid and the repeat constructs with the same restriction enzymes, so that I could then glue them back together essentially and make a new construct containing my my DNA, able to be targeted to the CSR locus. Then I transformed this plasmid, which is just a way of getting E. coli cells, or any cells for that matter, but in this case it was transformation into E. coli, so that the E. coli would reproduce and replicate the plasmid. And then I could harvest that DNA and later insert. But first I had to make sure that the insert was actually in there, because you can't see these plasmids with the naked eye. They're way too small, so you can't just like hold it up to the light and be like, oh yeah, of course. So I did what's called a gel screening, which is basically you take the DNA, that plasmid DNA you see in the middle there, you digest it with the restriction enzymes you see on the far left. That like cuts up the DNA into different segments, which you then run on a gel. And those gels cause smaller DNA to have like essentially less drag and run faster down the gel. So you see it further down at the bottom because of that. So it's a good way to separate DNA by size. So if it was positive for insert, we would see something like this. We'd see a big band of the majority of the plasmid and then one small region with the insert. However, if it was negative, we would have that same small region, but it wouldn't have the insert. And as you can see on the left, the actual gel, I've highlighted the one that was positive in green. And indeed, near the 700 base pair marker, we see a larger insert in one of the positives than one that was negative. And indeed, I found positive inserts for all of my strains with a good number of repeats, which was nice. And yeah, I just put that up there to have the corresponding, so you can see what bands belong to which. Next, I needed to get it inside my actual model organism, which is transformation into Neurospora. So first I get as before, the DNA to be taken up into Neurospora. And then it's inserted through something called homologous recombination, which you can see a general illustration of right here. So you have the plasmid DNA with insert that has been linearized, and the DNA kind of swaps with the wild type strain. So CSR1 gets replaced with the desired inserts, while the rest is killed. And this selection method is very convenient for making genetically pure cells, because Neurospora is multinucleic, and if a nuclei isn't successfully transformed, the CSR1, the presence of it will allow it to uptake a fungicide that'll kill that nucleus. So you only get Neurospora containing nuclei with the desired DNA insert. Or in some cases, if you have a mutation, you can get some false positives, but we're not going to talk about that. So this is a similar screening to what I did before with inserts into my plasmid, but a little bit more complex, because obviously we're not just dealing with a small plasmid right now, we're dealing with an entire genome of Neurospora. So this southern blot, or southern hybridization, allows me to just check a certain region of DNA for inserts. And in this case, I probed for the CSR1 locus. So I only get fragments that are around that region. So all the other DNA is just cut up and lost. And so you can see, like before, if I pop up the previous thing up there, you can see that it does look very similar to how it was before, and it is the same principle. Next, after I've confirmed that all of these repeats were successfully inserted into Neurospora, I did something called a chromatin immunoprecipitation. Now, basically, you've seen chip tracks before. I've put them up, or chip sequencing tracks rather. But basically, it's a way of, again, finding regions of DNA that have something bound to it. So what you do is you can see up there in the next to the word cross-link, a bunch of nucleosomes on a string. So what you do is you cross-link the DNA to the protein so they can't be separated. Then you shred the DNA that's not bound, so you are left with little fragments of DNA bound to proteins. You immunoprecipitate them, so you have an antibody, in this case, for H3K27 methylation, and then you basically wash out everything that's not bound to those antibodies. So we're left with only the DNA fragments bound to protein of our desired sequence. Then you reverse the cross-linking to release the proteins and purify the DNA, getting out those. Okay, it's telling me my meeting is going to end in 10 minutes, but I don't think that's true. I might have to make you the host again, Barbara, in just a second. But yeah, you purify the DNA, and then you can do certain things with it, such as chip quantitative PCR, which allows you to essentially count a certain section of DNA to see how many times it was bound to that protein, and chip sequencing, which shows you the entire DNA strand. So I did chip qPCR first because it is a lot more cost effective than other things, and I wanted a quicker result. And as you can see, we have my repeat strains minus the Arabidopsis and the fold increase of H3K27 over a part of the genome that doesn't typically have any. So this is just like how much it is enriched. And you can see in the delta set 7 strain, which should not have any H3K27 methylation, you see basal levels. And in the green there is my negative control for G quadruplex, and that shows statistically insignificant difference between them. So no methylation. And all the other strains that can form G quadruplex do show enrichment of H3K27 near CSR, both the 3' and the 5' regions. Interesting result. Now, the difference between qPCR, chip qPCR, and chip sequencing, or ChIP-seq, is that imagine you go to Google Maps and you want to know, how the traffic is on the highway. Chip qPCR essentially gives you just one section of the highway. Like, yeah, is there traffic on that section? Like, what do you guys think? I mean, maybe, or there's 3 cars that are like having fun just driving together, we don't know. It's hard to know how much traffic is going to be on your route, or if you're going to be late, But chip sequencing gives you the full picture, the entire highway. This is the information you want, because it gives you the context, right? So now we can see that there is indeed plenty of traffic on the road, or we would have found out that it was not as well. So this is chip sequencing of the non-native repeat strains. Up at the top, you have the wild type strain, so that is no insert at all. And as you can see, it's pretty basal in this region. You can see like maybe some things peeking out of background, but pretty much nothing. I have the CSR1 region where the repeats are labeled on the top right there. And the green track is the wild type strain used in the previous paper. So I just put that up there for a comparison to make sure mine was working. Right below that in the teal, you see my repeat strains, which show a very similar pattern of methylation to what was observed previously, which is a great sign. The negative control in the pink, TTA-GCG with no G quadruplex, you can see maybe a little bit of enrichment, but it's fairly baseline, and I would guess fairly similar to wild type, just you can see visually. TTGGGG, which is the tetrahymena, we saw robust enrichment, which was amazing, but also somewhat resembles the previous papers. Interestingly though, near the bottom, TTTAGGGG, the Arabidopsis, we did not see enrichment of methylation, which is peculiar because it is known to form G quadruplex in vivo based on previous experiments. So, why would that be? And well, I had a few possible mechanisms, because it's easy to include that the ability to form G quadruplex is not alone sufficient for internal repeats to induce ectopic H3K27 methylation. And one mechanism that I thought was quite likely was TERFs, or telomere repeat binding factors, could be involved, which they are known to bind PRC2 and also bind telomere and teleblock sequences, which are just smaller regions of telomeres found in the middle of DNA. And because there's an affinity for both of them, it might recruit PRC2 to a region for the methylation to be induced. And why this might explain it is because the binding pattern to the telomere repeats is incredibly specific. Like, it always happens in the same way, and you can see just the sheer complexity. There's like over 30 different binding interactions. So I was thinking that maybe the Arabidopsis, because it has 7 base motif of repeats, that might offset it eventually enough for TERFs to not be able to bind quite as well, and maybe that would affect things. Another mechanism that I thought could be responsible are telomere repeat RNAs, TERAs, which are also known to bind PRC2, and they are basically short RNAs that are transcribed from native telomeres usually. And I was thinking they could also be transcribed from internal telomere repeats that I inserted. So what that would look like was, as this sequence of internal repeats is being transcribed, you would have the RNA coming out of the polymerase, and if PRC2 were to bind to that, then that could temporarily get it to linger around the DNA that was being actively transcribed, which would potentially get it to do its catalytic activity there. It also has been known to associate with some TERFs, so there may be like, interaction between these two hypotheses, which in my opinion makes me trust it more. Some future directions, I think it would be good to insert these repeats into a delta TERT or a telomerase reverse transcriptase strain and do the same experiment once again, because if there's no native telomeres, then natural tera generation doesn't happen. So we can see if those patterns of methylation remain consistent even without native teras. Another thing that would be good to do is inactivate or knock out the telomere repeat binding factors in strains with inserted repeats to see if those TERFs are necessary for this induction of ectopic methylation. And something I would think would be good to do, but wouldn't necessarily directly do that, is to test Neuraspora crassa's like TERF binding affinity to DNA constructs, as I had on the last slide, just to see how, because they only did it for the native telomere repeats, the wild type sequence. So doing it for a variety of other sequences that I created would be a good idea to see if that would have any effect on anything. And yeah, I'd just like to, first of all, thank my mentors, Eric Selker, of course, and the rest of my committee, Barbara Mossberg and David Garcia, for being with me along this process. My direct mentor, Kevin McNaught, I don't think he's here right now, but he got married. He can do whatever he wants. But he was a great guide on this project. The entire Selker lab as well. And yeah, any questions?